Hey learners, welcome back to the part 2 of this chapter 8, Cell, the Unit of Life. So the topics to be covered in this chapter are as follow, and as I said, this is the part 2. We are already done by few of the topics like what is a cell, cell theory and prokaryotic cell. In this lecture we'll see an overview of cell, a brief comparison between both the prokaryote and eukaryote cells, and we'll discuss eukaryote cell in detail in this lecture. So let's begin by this. So we'll look by the differences. First we have prokaryotic cell. Prokaryote means pro means the primitive one. Karyote means nucleus. So prokaryotic cells, they are smaller in size, about 0.1 to 5 micrometers. Then we have eukaryotic cells. They are more complex and modified form of prokaryotic cells. They are larger in size, about 10 to 100 micrometers, and prokaryotic cells are always unicellular, and eukaryotes are unicellular or multicellular both. If you remember the 5 kingdom classification, Protista comes under the unicellular eukaryotes, while other 3, fungi, plantae and animalia, they comes under the multicellular eukaryotes. Prokaryotes comes with the false nucleus. It means it is not nucleus in real, but it only look like the nucleus. That is, the more appropriate term for it is nucleoid, the region in which it lies, their chromosome lies, and it is often called as incipient nucleus, brilliant nucleus or fibrillar nucleus. These terms are really old and less commonly used for the nucleoid. Scientists used to call this primitive undeveloped form of nucleus as incipient nucleus. It is also called as brilliant nucleus. See, once you look under microscope, you see this thread or fiber-like appearance of DNA in the nucleoid region, and that's where it gets its name. And again, they were called as fibrillar nucleus because of their appearance, their texture, thread-like fibrous DNA texture. That led the scientists to call it as these names, but in the correct term for the modern biology is nucleoid only. On the other hand, eukaryotes have a well-developed double membranous nucleus, and other than nucleus, if we talk about DNA, these prokaryotic DNAs, they have the single number, only one double standard circular DNA, which is naked in nature. I have already explained their double standard circular structure in the previous lecture, and similarly in the eukaryotes they also have this double standard DNA, but the only difference is they are linear in structure. Although their DNA is packed with the histone protein, but in the prokaryotes their DNA is naked because it is not packed in any histone protein, but it is in place of the histone protein, polyamines are present. And because of all of these characters, these prokaryotic chromosomes are also called the false chromosomes, like their nucleus, but in the eukaryotes they have very well defined chromosomes present there. And other than that, of this nucleoid or main genomic DNA, there is one more extra chromosomal piece of double standard circular DNA is present, which is called as plasmid. It's not a conformational character of these bacteria, but their presence varies upon the type of bacteria. The prokaryotic cell wall is made up of peptidoglycan. They are present in different diversity among them. With the exception mycoplasma, it lacks the cell wall. While in the eukaryotic cell, their cell wall is made up of by the different compositions in different organisms. Like in the fungi, they are made up of chitin, and in the plants, their cell wall is made up of cellulose and hemicellulose, pectin, proteins, and in the algae, their cell wall is composed of cellulose, galactose, mannose, and minerals like calcium carbonate. And if you notice that their all cell walls are composed of the structural polysaccharides, right? And in the eukaryote, cell wall is not a permanent character. In the plants they have the cell wall, but in humans they don't have any. But in the prokaryotes it's a defined, well defined characters for them. And if you look in the prokaryotes, you will find that there is no compartmentation is present. It means their membrane bound cell organelles are absent in the prokaryotic cells, while in the eukaryotic cells there is an extensive compartmentation is present there in the cytoplasm throughout it, in the presence of membrane bound cell organelles, where different types of work done by different organelles, of course. And the locomotory organs like flagella, they in prokaryotes they are made up of flagellin protein with very simple structure, and in the eukaryotes they are made up of microtubules with the 9+2 arrangement, tubulin protein you can say more specifically, and they are comes with a very complex structure in compare with the prokaryotes. And by moving towards their common cell organelle, which is ribosome protein factory, which is in the prokaryotes they are 70s ribosome, and in the eukaryotes they can be 70s or 80s both. And as I said, prokaryotes are unicellular, so they can only perform asexual reproduction by the binary fission. You may know that's a very basic one. And in the eukaryotes, they can be unicellular or multicellular. They can be both, so they can perform both the functions accordingly. If they are unicellular, they will perform asexual reproduction, and if they are multicellular, they will perform sexual reproduction by meiosis and mitosis. And as we know, in eukaryotes there are always organs defined for the activities which they perform, like for respiration it is mitochondria, but in prokaryotes they don't have any cell organelles, so they respire through the mesosomes. They can perform both aerobic and anaerobic respiration in prokaryotes, while in the eukaryotes aerobic respiration is prime to them. Only some cells can perform anaerobically in eukaryotes. So their ATP forms in the cytoplasm, basically inside the plasma membrane, and in the eukaryotes their ATP forms in specifically in mitochondria. And there is another extension of plasma membrane in prokaryotes which is called chromatophores, where all their pigments are stored for their photosynthesis, as you can see in the image. But in the eukaryotes they have chloroplast for their photosynthesis. Now we will see the types of eukaryotic cells. So they can be classified on the basis of their structure, function and their kingdom which they belongs from. But broadly we'll classify them in only two types, which is plant cell and animal cell, and we'll see their differences also. Human, dog, fishes are the example of animal cells, and leaf cells, plant root cells, stem cells are the examples of plant cells. So while we look by the features of them, so you may have earlier observed cells in an onion peel and human cheek cells under the microscope. Let us recollect their structures again. And the onion cell, which is a typical plant cell, has the distinct cell wall as its outer boundary. But in the animal cell, for example, the cells of human cheek, they don't have any cell wall out of it. Then centrioles is the characteristic of animal cells only. They are not in the plant cell, neither in the prokaryotic cells. And the animal cells don't have plastids in them like the plant cells, because they don't need to perform photosynthesis for their food and other. Similarly, animal cells don't have vacuole because their structure is more dynamic and flexible. They eat frequently, so they don't need a big permanent storage space like vacuole inside their cell as the plant cells do. Other than that, in animal cells they store their extra food in the form of glycogen, and in plants they store their extra food in the form of starch. You already know that. So these 4 to 5 are the major differences between them which you need to remember. Now this is a diagram showing comparison of some eukaryotic cells with the other organisms of the prokaryotic cell. We'll see the direct comparison by the shape and sizes. While a typical prokaryotic bacteria is of 1 to 2 micrometer, and their smallest cell PPLO, which is of 0.1 micrometer. Even if we talk about viruses, which is neither prokaryote nor eukaryote, even they are of 0.2 to 2 micrometers. And if we compare these prokaryotes with a typical eukaryotic cell, they are about 10 to 20 micrometers, which is almost 10 times larger in their size, shape and volume as compared to the prokaryotes. And as we know, the smallest prokaryotic bacteria is Mycoplasma PPLO, and the biggest bacteria known till the date is sulfur pearl, which is found somewhere in the oceans of Africa. They can be seen by your naked eyes also. This bacteria actually breaks all the rules. I mean, who knows, bacteria could be so big to be visible by the naked eyes. And in eukaryotes, the largest isolated single cell is the egg of an ostrich. I mean, they can be, of course, seen by the naked eyes. And the fun fact about it is the entire yolk of the ostrich egg is actually one single cell. And the smallest animal cell is of male bee, whose length is equal to the length of virus. It means they are extremely smaller. They are smaller than the many of the bacteria. And if we talk about humans, their biggest cell is ovum, egg cells, which is present in females. They are visible without a microscope, and the smallest cell is sperm cells in the males, which is very small. But I would like to tell you, in general, sperm cells across all the animals like insects, mammals, humans are among all of them. The smallest animal cells are these sperm cells. Then let's move to the different shapes of the eukaryotic cells. Cells also vary greatly in their shapes. They may be disc-like, they may be polygonal, sometimes columnar, sometimes cuboidal. They can also be thread-like, or even up to irregular shapes are there. The shapes of the cells may vary with the functions they perform. So we'll see, among all the multicellular organisms, we have few of their organelles on this slide. Like human red blood cells, they are about 7 micrometer in their diameter with round and bioconcave shapes. Then we have nerve cells. They are one of the longest cell in our human body, which comes with the located in our brain, which comes with the long and branch shape. Also we have white blood cells with the amoeboidal shape, means they change their shape like an amoeba. Then we have epithelial cells, which are columnar, long and narrow in their shapes. Then we also have some of the plant cells to be discussed with the no cell membrane in them. They are more like dead organelles. So one of them is tracheid, which is elongated in shape, xylem cells. And then we have mesophyll cells. They are the leaf cells, which are round and oval in their shapes. Then we will see cell wall, which is a plant specified characters. They are dead, rigid and permeable in nature, where the permeability means they allows all the solute and solvents to pass throughout it. They are called dead because they don't carry out any living activities like metabolism, respiration and transport. They are made up of non-living materials like cellulose, and also they are the rigid structure because they are made up of the strong structural material cellulose in them. You may recall this cell wall forms an outer covering of the plasma membrane of the 5 kingdom of classification, where Monera consists of peptidoglycan in bacteria, and in the protista they usually don't have any cell wall. Sometimes they have, sometimes they don't. And in the animalia they don't have any cell wall, we know this. And after that, mainly we will focus on algae and plants, which belongs from the plantae kingdom, and fungi. So let's begin by this. In fungi, you already know that they are made up of chitin, which is a polymer of NAG plus NAM. And in the algae, they are mainly composed of cellulose as the main component, then galactans as the polymer of galactose, with mannose, which is a polymer of mannose, and minerals like calcium carbonate. Then we'll also have an exception of euglenoids, which belongs from the protista kingdom. They don't have a true cell wall. Other than that, we have plants with cell wall is composed of cellulose, hemicellulose, pectin and proteins. We'll see their structure in detail by now. So let's move by the next slide. The cell wall is divided into 3 main layers. If you see the cell wall of a young plant cell, you will find that these 3 layers, the first one is primary wall, then you will find secondary wall, and there is a middle lamella. The primary cell wall is thin and elastic in nature. See, it is obvious. The primary cell wall is formed when the cell is inactively growing. So it must be flexible and thin so that it could allow the cell to increase in size to absorb the water. These thin walls also helps in the easy diffusion of water, gas and nutrients between the cell, and their elastic nature lets the cell wall to stretch without breaking. So the matrix of the primary cell wall is like a soft jelly where the microfibrils are arranged in a crisscross manner, where you can find like each layer has its own microfibril arrangement. As you can see in the image, these microfibrils actually tries to hold all these thread-like cellulose structures together in a proper way. But what if these microfibrils are disarranged in any ways? Then these complete cell wall will be breaked out. Suppose you plays basketball, right? You hit the ball in the net so hardly so that all the its net got randomly loosen out, and the basket may still not hold things. It can collapse under the weight, right? Similarly, now they are composed of cellulose, hemicellulose, and pectin. You know these compounds, right? From the biomolecule chapter. Cellulose is a homopolysaccharide while hemicellulose is a heteropolysaccharide. Cellulose provides the basic strength and rigidity to the cell wall. It is more like the steel rods in a concrete, they gives the wall its basic strength. While the hemicellulose tries to bind these cellulose microfibrils together by providing them support and flexibility, like a glue or connector tries to hold the steel rods together. Now here comes that gel-like substance which we're talking about is pectin. It's a gel-like polysaccharide, highly hydrated, soft and sticky in nature. It retains all the water in the cell wall. It's like a softener, wall softener, and it's like a jelly or that cement that sticks brick together and makes the wall soft yet connected. And your NCERT says that these primary cell walls are capable of growth, which gradually diminishes as the cell matures. Now we have secondary cell wall. These are found in some mature cells only. They are located beneath the primary cell wall. They are actually formed on the inner side of the cell towards the plasma membrane, of course. They are thick and rigid in nature. As they have no role in growth, they don't need this flexibility in such terms, right? So they are thick and rigid. Also, they don't store water in them to give it more rigid structure. And their matrix is composed of cellulose, hemicellulose, pectin, and there is one another new thing which is lignin. Now we'll see them. Even if you see in their matrix, you will find that secondary cell walls contain more cellulose than the primary walls. These cellulose microfibrils are even tightly packed and arranged in multiple distinct layers. As you can see in the image, S1 is outer layer, S2 is middle layer, and S3 is the innermost layer. At all the different angles to making this structure super strong and durable. If we talk about hemicellulose, they are again more in the secondary walls, but the pectin is little less. And last but not the least, we have lignin, which is a complex, rigid, non-carbohydrate polymer. They are found mainly in the secondary cell walls of the plants. It is not at all flexible like the others. It makes the cell wall harder and waterproof. And these lignin are the main reason for the hardness of the wood. You can remember it. Lignin is more in the secondary walls while they are absent in the primary walls. On the other hand, pectin is more in primary walls, but they are almostly absent in the secondary walls. Rest of the things are similar in both of them. And don't forget, they are only present in the mature cells. After all, once the growth is complete in the cell needs protection and more strength, not the flexibility like the primary walls, right? Then we have middle lamella as the outermost common layer between these two adjacent plant cells, composed of pectin, sticky substance pectin. It consists of calcium and magnesium pectates within it, a gel-like cement. That is why it is also called as plant cement. So when it binds with these calcium and magnesium ions, it creates a sticky glue-like structure to hold all the cells together. When remember, these calcium pectates are always more in number. Although middle lamella is not a true wall layer, but it is considered as one because it was the first formed layer as discovered as the first form layer, but it functions as the cementing material between the adjacent plant cells. So the middle lamella is technically both a cell wall layer and also it acts as a natural cell junction, unlike the other animals who uses tight junctions like desmotubule, but in the plants they rely on the middle lamella for their cell to cell adhesion. Okay, now let's take this through a practical point of view. Can you answer me? Like whenever we try to eat raw fruit, it become a little difficult to do so. But once it get ripen up, it's easy to have the fruit. Can you answer me why? What is the reason or factor behind this thing? See, we all know that middle lamella is full of calcium pectates that make it tough. But at the when the cell is unripened or raw, the enzymes that break down these pectins are at inactive stage. But once these fruit get ripen up, these pectinase enzymes get activated, and the enzymes tries to break down these pectins, especially in the middle lamella. By breaking these bonds between these hard calcium magnesium pectates into soluble jelly like pectins. And suddenly the middle lamella starts dissolving, the cells get separated slightly, making the fruit more softer. Why it happened? Because the starch turns into the sugar by increasing the sweetness. That is the reason why the ripened fruit becomes soft, juicy, sweet and edible to eat. So yeah, that is the conclusion why middle lamella is a key reason we unable to eat the unripened fruits and we able to do so when the fruit get ripen up. Then we have Plasmodesmata. This name was proposed by Strasburger in 1901. Even the term cytoplasm was also given by him only. We will explain it through this diagram of Plasmodesmata showing how two plant cells are connected with each other through their cell walls. So now the question is, what is plasmodesmata? So these plasmodesmata are tiny channels with the bridge-like structure that passes through the cell walls of the plant cell by connecting the cytoplasm of one cell to the another cell. That allows the communication and transport directly within the cells. Your NCERT says that the cell wall and middle lamella are traversed by the plasmodesmata, which connects the cytoplasm of the neighboring cells together. If you look deeply inside the structure of plasmodesmata, you will find there is another tubules or desmotubules are available which help to maintain the continuity of the cytoplasm. It means that's the tube that drives from the endoplasmic reticulum that runs throughout the center of the cell. Okay, let me clear this with simple analogy. Suppose this is my house, house A, and this is your house, house B. Okay, think of the plasmodesmata like the secret underground tunnel between these two houses, which allows people of the house to walk from one house to the another without using the road outside the extracellular space of these two cells, or you can say these two houses, where these desmotubules are like pipe inside the tunnels which runs from inside of the tunnel uses for to carry specific things like packages, signal, electricity, water and many such things. That is the reason why cell use these plasmodesmata and desmotubule structures to stay connected and share resource with each other quickly and directly. So the moral of the story is stay connected with your neighbors. Don't ever fight with them. You never know when they will be useful for you. Now we will see the functions of the cell wall. So first it gives the shape and structural support to the cell. It means the cell wall determines the shape of the cell. Then it provides a strong structural support to prevent the bacterium from bursting or collapsing. Yes, because of the hard structure of the cell wall, it is always in some specific particular shape. Cell wall is a dead and non-living structure, so it doesn't really perform any specific function related to it and its metabolism. So it provides protection from the injuries like the mechanical damage and pathogenal attacks like the infections, and the cell wall helps in the cell to cell interaction by the structures like plasmodesmata which we just saw. and it provides barrier to the undesirable macromolecules. You already know that it is a permeable layer. It means it allows all the solute and solvents to pass throughout it, but not the big macromolecules, right? And at the end, the cell wall prevents the cell from the cell bursting. Now suppose we have two beakers filled with water, which is named as beaker A and beaker B. So in both of them, we'll take the example of bacteria. There is one bacteria in each of them. So in the beaker A, there is a bacterial cell with only cell membrane, and in the beaker B, we have the another bacterial cell with the cell wall and cell membrane together, both. Now we will see what's going on inside the beaker. So these water starts enter in the plant cells by the osmotic pressure, and it creates the turgidity inside the cell. That is a pressure which pushes the membrane outwards, and suddenly the cell bursts. But in the another beaker, you will see the cell has another layer of cell wall on top of the cell membrane that resists the pressure created by the water intake. Now I hope it is clear to you that how these cell walls prevent the bacteria from bursting. Now the plasma membrane, also called as plasma lemma, cell membrane and biomembrane. Their structure is universal in both of the prokaryotes and eukaryotes. They are structurally and functionally they both are similar, even though they can be chemically similar but with different composition. For the examples, there are few bacteria that can survive in extreme temperature and also in the lowest temperature. They do have some special adaptation in the plasma membrane, which we'll see further. First, we will see the basics of cell membrane, like the discovery and composition. So let's begin by this. So the detailed structure of cell membrane was studied only after the advancement of electron microscopes in the early 1950s. Meanwhile, the chemical studies on the cell membrane, especially in the human red blood cell RBCs, enables the scientist to deduct the possible structure of the plasma membrane. These studies shows that the cell membrane is mainly composed of lipids, proteins and carbohydrate. Their general cell composition of protein is 52%, lipid is 40% and carbohydrate is about only 8% in it. Now many of you were wondering why the scientists used only RBC for their chemical analysis. Is there any reason behind this? Yes, they did it on the RBCs because the mature RBC lack all the cell organelles. I mean, there is no ribosome, no centriole, no mitochondria, no nucleus, and no DNA, no any endoplasmic reticulum. There is no such organelles are there. This means only the plasma membrane surrounds their cytoplasm, making it easier to isolate the plasma membrane and study these membrane without any interaction or interference from these any other internal organelles, as I said. That was the main reason behind doing this. Other than that, RBCs are the most abundant and easily available cell in the blood. We know that, right? So scientists can collect these in large quantity of RBCs from the same sample or same blood sample and get pure membrane samples out of it for their analysis. So earlier only phospholipids are known to be in the cell membrane. Later on, the other biochemical investigations clearly revealed that the cell membrane also possesses protein and carbohydrates in them, and their ratio of protein and lipids varies considerably in different cell types. Other than that of these erythrocyte cells, if we talk about the general cell composition of where the protein is about 60%, lipid is about 40% and carbohydrate contains 1 to 2% approximately. Now we will see the structure of cell membrane. Their structure is described by the fluid mosaic model, which is given on your screen. So this model is proposed by Singer and Nicholson in 1972. This is the latest and the most widely accepted model for the structure of the cell membrane. So according to this model, it says that the proteins are arranged in a phospholipid layer as the mosaic pattern. Thus, these membranes is termed as the protein iceberg in a sea of phospholipid, or you can say gulab jamun in a concentrated solution of sugar. where the gulab jamun is compared as the protein which floats in the sugar solution, which is compared as phospholipid. Similarly, you can say gulab jamun as iceberg floating in the sea of phospholipid. Okay, so now let's move by their components. First, we'll see lipids because they acquire 40% part of them. So let's begin by it. Now, the study says that the cell membrane is composed of phospholipids that are arranged in a bilayer. As you can see in the image, if we take a small section or the part of cell membrane, we will find that there is a phospholipid bilayer, and if we look deep inside them, we'll take a single phospholipid out of this bilayer structure and we will examine what's inside it. So, as you can see in the image, first we have this polar head of the phosphate molecule which faces the outside environment, and they are hydrophilic in nature, means they are water loving, and as they are water loving they are also lipophobic in nature, it means they are fat repelling. Okay, and then we have these non-polar fatty acid tails which faces the inner part of the cell, inner environment, and they are hydrophobic in nature, means they are water fearing, they repel the water, and as they hate the water, they are we can also say they are lipophilic in nature, means they love lipid. Now it is very obvious that if there is a compound which is hydrophobic in nature, then it cannot be lipophobic at the same time. Why? Can you answer me? Yes, because of their polarity. We know that water, and what are these? Water and these lipids, what are these? These are just the solvents. So water is a polar solvent while the lipid is a non-polar solvent, and we know that like dissolves likes. It means that the polar dissolves with the polars only, so it will repel with each other. So these polar and non-polar substances do not mix with each other. So the phosphate head avoids the lipid environment and prefers the water side. And these lipid tails avoid the water environment and prefers the lipid side of the cell. Right? And if you can see it, this is what I was talking about, the intracellular fluid. It means the extracellular part of the cell, which is water, and this is the cytosolic fluid, which is the intracellular, or you can say internal matrix of the cell, which is almost about 99% is water, where the amphi means both. Oh my god, don't be confused with the amphoteric compounds which was in the biomolecules, where the amino acids is amphoteric compound. It means that it contains one acidic COOH and one alkaline group NH2. They both are different things. Amphipathic molecules are water loving and water fearing molecules. They are not related to charges at any cost. Other than that, these arrangement creates a semi-permeable barrier towards the outer biomacromolecules, which allows the only the selective entry and exit of these substances. Now we will see by the fluidity factors of the cell membrane, which depends on the fatty acid. See, we know that in the phospholipid bilayer of the cell membrane, there is one hydrophilic head with two hydrophobic fatty acid tails, and these fatty acid tails of these phospholipids can be either saturated or unsaturated fatty acids. You know all these terms, right? And this differentially greatly and influence membrane fluidity and factor in such ways. So in the saturated fatty acid, there is only single bond present, and with no straight chains. As you can see in the image, there are saturated hydrocarbon tails. They are straight chains, and their tails pack tightly. It's clear in the structure, and their membrane become more rigid and less fluid because of their tight tails. And in the unsaturated fatty acids, they could be of single and double bonds, and with a kink, they creates a kink in their chain. You can see in the structure of unsaturated hydrocarbon tail, they are with kink, which is kink is something you can relate with knot, as they are anti-parallel structures of their tails. Okay, these tails do not pack tightly because of their kink structure, and because of that these membranes become more fluid and flexible as compared to the saturated one. So phospholipids has one of each, but across the entire membrane, unsaturated fatty acids are slightly more to ensure the fluidity as compared to the saturated one. So basically, the cell membrane adjusts the ratio of saturated to unsaturated fatty acids depending on the temperature and environment to maintain the optimal membrane fluidity. Now the question arises here is how these unsaturated fatty acids maintain the fluidity of the cell membrane. See, for the very first thing is these unsaturated tails prevent the tight packaging of the cell membrane to keep their membrane more fluid and more flexible by maintaining their quasi-fluid nature of the cell membrane, and which is essential for protein movement, cell signaling, and transport of the things materials more easily. And that too, in the human cells they are about 50 to 60% in composition, and rest 40% is saturated fatty one. Now we will see how temperature and cholesterols affect these fluidity and their nature. So let's begin by them. Other than that of phospholipids, there is one more lipid which is present in the plasma membrane, and that is cholesterol. So cholesterols are more rigid and than in compare with the phospholipids. So it helps in the stability of the membrane, cell membrane in their structure. Now, can you answer me why cholesterol has this rigid structure? Yes, because of its structure. Cholesterol is a derived lipid. It means it's not a true lipid. They are composed of the fatty acids, which is a simple lipid. Unlike the other regular lipids, cholesterols don't contain such long fatty acid chains. Instead, it has a ring structure, which is why it is not fluid or flexible like the fatty acids, but it is rigid. And this rigid structure makes cholesterol solid and non-fluid in nature, unlike the freely fatty acids or phospholipid tails. And if you see their arrangement, these cholesterol molecules are embedded between these phospholipids in both of these layers. It fit partially in the head region and mostly in the tail region, as you can see in the image. Now suppose each phospholipid is like a soft pillow. Now what if I insert a hard book of cholesterol between these pillows? This hard book will keep them either sticking too tight or spreading too loose. Now again, like the phospholipids, these cholesterols are amphipathic in nature. It means they are consist of one hydrophobic and one hydrophilic part. The hydrophilic part is hydroxyl group, OH group, you can see there in the chemical structure, and one hydrophobic group which is steroid ring structure. Now we have the another term for cholesterol, which is cholesterol buffer. It means it's a special unique ability of the cholesterol to maintain the membrane's fluidity by keeping the membrane neither too rigid or nor too fluid. That depends on the temperature. By stabilizing the fluidity of membrane across different temperatures, just like a thermal insulator do, it prevents the extreme changes in the membrane state. Now suppose in the cold temperature, phospholipid layer packed tightly and the membrane become too rigid. But in vice versa, in the hot temperature, these phospholipid layers move too freely and these membranes become too fluid. They are not too stiff, not too leaky, so that the membrane stays stable and functions across varying conditions and temperatures. So the membrane fluidity and membrane integrity both are maintained by the cholesterols only, regardless of their temperature, whatever it is, either highest of 40 degree or lowest as 10 degree. And despite of them, and yeah, I forget to tell, this is the characteristics of humans only, but in prokaryotic cell membranes cholesterol is absent there, but in place of the cholesterol there is a hepenoid present in them, which controls the fluidity of this prokaryotic cell membrane. Of course, we're taking the exception mycoplasma. Even in humans, we are homeothermic organism. It means we are warm-blooded, able to maintain a stable internal temperature, which is constant 37 degrees Celsius. Still, we need cholesterol buffers to maintain the fluidity buffer, because the local organs and different tissues and cells and organelles can vary in their structures and functions. Although we don't need to go that deep inside of these topics, so let's move by the next slide. Now we will see the actual mosaic pattern, the protein part of the cell. So these proteins are embedded in or attached to the lipid bilayer of the cell membrane, and they can move literally like a boat floating in a sea of the lipid. So as I said, there are two types of proteins and their structures are present in the cell membrane on the basis of their age of extraction. Okay, so the first one is integral and also we can say intrinsic proteins. Then the second one is peripheral and extrinsic proteins. So let's begin by them. So first we will talk about the peripheral and extrinsic proteins. The name itself says that peripheral means they are on the periphery of the cell membrane, or the extrinsic proteins, it means they are on the outer side of the cell membrane. And as they are loosely attached to the outer surface of the cell membrane, they can be easily extracted as well. And if you see, they are bounded to the integral proteins, or you can say the polar heads of the phospholipids. And if we talk about their functions, we already know that some of the proteins acts like an enzyme. So these are the one who perform enzymatic activities. And these peripheral proteins also play the crucial role in cell signaling and the maintaining the cytoskeleton structures by acting as connectors and messengers or the structural anchors just beneath or outside the cell membrane. In the multicellular organism, cells must be able to communicate with one another by sending messages by the cell phone, and to do that, cells send out these tiny signals that act on the receptor of the other cell. They are like, hey cell, can you hear me? Is the signal is coming out there? Like something like that. And this distance between this signaling cell and the receptor cell is called as the signal. And they also help in transmit the signals from outside to inside of the cell and vice versa, inside to outside of the cell, by working with the integral receptors and secondary messengers. Then we have integral or intrinsic proteins. As the name says, they are on the inner side of the cell membrane. Also, they are tightly embedded within the molecules of cell membrane. And because of that, they can be of two types. The first one is they can be partially embedded, and they can be completely embedded. Partially embedded or the monotopic integral proteins are inserted only into the one side of the bilayer structure of the cell membrane. They are often involved in the enzymatic or signaling roles. For example, we have prostaglandin synthesis, which is involved in the inflammation of. Then we have completely embedded proteins, which is also called transmembrane proteins, because they cross the entire membrane of this phospholipid layer. If we look by their functions, we'll see they help in the transportation of the molecules. For example, we have different structures specialized for them. For ion, we have ionic channels. Then we have carrier protein, and we also have channel or tunnels. They also serve as receptors for cell signaling molecules, like example hormone, insulin, and receptors. You can clearly see in the diagram that the hydrophobic region is within the bilayer and the hydrophilic region is outside of this cell membrane region. Now we will look by these transportation system, their diagrams, their examples in more detail. So let's move by the next slide. Now we have this whole transportation system in a flow chart, where we will see the movement of molecules, substances like ions, water, gases in and out of the cell throughout this plasma membrane. We know that these cells are selectively permeable. Only specific substances can pass throughout it depending on their size, charges, polarity and needs. So on the basis of that, we have two main types of molecule transport on the basis of their size. First we have small molecules which is transported by the specific transportation system, which we will discuss about in this chapter. And then we have large molecules which is transported by the bulk transportation system, which we don't need to cover in this chapter, but still we'll discuss in the last. But first we'll start by transportation for the small molecules, which is further divided into active and passive transport. In the passive transport, we will see simple diffusion with the examples like osmosis, respiration, and in the facilitate diffusion, we will see carrier protein and channel proteins. Then we have active transport, where we will discuss about pump proteins. So let's begin by the passive transport. Then we have passive transport. So the passive transport is a transport where the cells pass throughout the membrane without any energy. They don't need ATP, they don't require any energy needs, and the substances move from higher to the lower concentration along the concentration gradient. It is very obvious that moving from the higher to lower among the concentration gradient, you don't need any energy, right? We know that. And further they are subdivided into few types, which is simple diffusion, osmosis, where the simple diffusion is the movement of small non-polar molecules directly through the lipid bilayer. For example, the diffusion of oxygen and carbon dioxide in the lungs. Then we have osmosis, where the movement of water molecules take place across a semi-permeable membrane. For example, we can say the movement of water molecules in the plant roots. Now look at this diagram. These are the small uncharged molecules like oxygen, carbon dioxide, nitrogen, glucose, and many more. So these are passing freely in through this plasma membrane. And these are the extracellular fluids which are outside the cell membrane, and we have cytoplasm inside the cell. Then these molecules are initially more concentrated in the extracellular space, as you can see, and over the time the concentration of molecules become more balanced on both of the side of membranes. You can see there is a timeline given on the bottom of the screen, which indicates that the diffusion down the concentration gradient become high to low, and at the end all of these small molecules are equally distributed on both of the sides, and cell reach its equilibrium without the use of any such special structures. And then we have facilitate diffusion, where we will see the movement of large and polar molecules like ions, sodium, potassium, and for that we'll need some special proteinaceous structures. You can imagine by yourself, as they are larger molecules, so it becomes kind of impossible to pass throughout it without the use of any of such special structures. And as their molecules are hydrophilic in nature, like the glucose or ions, so they can't pass freely without it, so they need this transportation protein structure to help them across these membranes. Now the channel proteins and the carrier proteins are the two main type of structures which are involved in their transportation. So let's see what are these. First, these channel proteins form the pores that allow the specific ion or water to pass throughout it. For example, we have sodium, potassium, and aquaporins. These aquaporin channels allows the water molecule to pass quickly across the cell membrane through their tunnel structure. You can see their tunnel shape structure in the image, and see, we need these structures because the water can diffuse slowly through the lipid bilayers, and these aquaporin, these specialized structures, speed up the process up to 3 billion molecules per second. Can you see the difference? Even at the molecular level, yes, I agree with you, they don't need any special energy, ATPs, but they take our time. So the aquaporins just facilitate these simple diffusions in an easy way. But yes, they can still diffuse through those lipid bilayers, but only very few of them, right? Then we have carrier protein. These are the proteins which bind with the specific molecules on one side of the membrane and undergoes a conformational change and release that molecule on the other side of the cell membrane. This process can involve in the change of shape. Tell me, isn't it interesting? Yes, it is. Okay, they are also highly specific towards the molecules they transfer, right? Where we have this example of GLUT4, which stands for glucose transportation type 4 of the glucose molecules into the muscles and the fat cells in response to the insulin, sorry. And yeah, if you compare it with these simple diffusions, these facilitate diffusions are a little bit faster because of that special proteinaceous structures. I hope it's all clear now. So let's move by the active transport. Unlike the passive transport, active transportation is quite different from them. In the active transportation, they requires energy in the form of ATP from moving against the concentration gradient from the low concentration to the high concentration. For example, we have sodium potassium pump. Its special ability to maintain the ionic balance by absorbing the nutrients even when the concentration gradient is low. That's interesting. By removing the waste from the inside of the cell, one of the famous example is pump protein. As the name says, it pumps and pushes the molecules across the membrane, similar to how a mechanical pump works. Now imagine a water pump. We use it in our home to move water from a lower tank to an upper tank against the gravity. It takes effort and energy to do so, right? Or you can say cycle pump. There you also applied some amount of energy to fill your tires. For example, we have sodium potassium pump. It's more like a two-way cycle pump. It works on the count of 3 to 1. Okay, let me explain it. First, it binds with the 3 sodium ions from the cytoplasm of the cell. Then it uses the ATP, change its shape and releases the 3 sodium ions outside the cell. Then it binds with 2 potassium ion from outside the cell and return to its original shape and releases it inside the cytoplasm. All by using only one molecule of ATP. Let me repeat it again. 3 sodium ions out of the cell, 2 potassium ions into the cell by using only one ATP. Is it clear now? Now we are done by all of these specific transports for the small molecules, and we also covered their active and passive transportation system. Now we will move by the bulk transportation for the larger and big molecules, where it's passive transportation, where we will see their structure, types and examples. So let's begin. Now, as I said, bulk transportation is used for the movement of large particles or the large volume of fluid or molecules across the cell membrane using the vesicles. So they are of two types, endocytosis and exocytosis. So first we will discuss endocytosis into the cell. So in the endocytosis, it's a process by which the cell membrane folds inwards to form a vesicle like structure that required some amount of energy, ATP units. That's why I said they are the one of the passive transportation system. So they forms this vesicle that brings the substances inside the cell, and there are two types, phagocytosis and pinocytosis. It's a process where cell eats solid substances, or you can say the membrane engulf all the larger and solid particles throughout the cell. This process also become crucial for the immune defense system and removing damaged cell and other external products for the tissue and other overall cell development. Then the pinocytosis, which is also called as cell drinking, where the cell dissolves all the liquid substances and fluid substances from the surrounding. This process also becomes important for the transportation of fluid, dissolved nutrients, and even for certain drugs across the cell membranes. Then there is another term which is called as exocytosis, which occurs out of the cell. This process is where the vesicles form inside the cell and fuses with the cell membrane to releases the substances out of the cell. There are of two types, which are excretion and secretion. So let's see them. Okay, so the excretion is the process by which a cell removes the metabolic waste and unwanted byproducts of the cell from its internal activities. These may be any unwanted substances, they could be toxic in nature, so they don't serve any functions out of this cell, and they do it by the process of diffusions also. If you look at their examples, we have carbon dioxide releasing during the respiration process, then we have ammonia in the unicellular organism, lysosomal waste released out of the cell by the process of this excretion. Then we have secretion, which is the release of helpful substances out of the cell, where it's a process where a cell releases substances or molecules to perform a specific function out of the cell. Their secretory materials are often proteins or enzyme. That's why they are helpful in the function of digestions. And we have some other examples similar to it, like the salivary gland secreting amylase, then pancreatic cells secreting insulins, neurons secreting neurotransmitters like dopamine, and some others. Now you can break down this diagram on the basis of your understanding. Okay, let me do it for you real quick. You just focused on the screen, right? So suppose this is a white blood cells engulfing the bacteria. On the top of the diagram, it's written there, it's phagocytosis, cell eating, where the cell engulfs this solid part, the bacteria, from outside the cell, where these vesicles travels inside the cell. Eventually, they fuse with a lysozyme for their digestion. So, in depth, their purpose is to destroy the harmful materials. Now look on the bottom of the diagram. Pinocytosis is written there. Now suppose it's an intestinal cell which absorbs all of the extracellular fluid from outside the cell, and then these fluids liquid dissolves in the solution of the cytoplasm, and they fuse with the lysozyme. The absolute purpose is to absorb these fluid nutrients. Then look on the right side of the diagram, there is two type of secretions given there. One is vesicle related secretion and another is lysosomal secretion. So despite of what secretion they are doing, we just need to know what they are doing, right? So let's not go that deep into it. Now suppose these vesicles are filled with the useful substances like enzymes, hormones, and etc. Then they fuse with this membrane released contents outside. Then these lysosomes, we have lysosomes, they are released with some digestive enzymes out of the cell. Then at the end there is excretion, waste removal process seen in the leftover vesicles are broken down the product after digestion by lysosomes. These waste vesicles also fuse with the cell membrane and they are exposed out of the cell. Their purpose is waste disposal, not to perform any extra other functions, right? Now let's move by the next slide. And yeah, just don't focus on the extra other things given on this diagram. I have already explained all of the important things. Now let's begin by the carbohydrates. Then we have carbohydrates. Carbohydrates plays several critical roles in the cell membrane. Even though they are the only small part of its structure, they are superficially arranged on the surface of the outer surface of the plasma membrane. So when they are attached on the protein's structure, they are called as glycoproteins, and when they are attached on the lipid structures, they are called as glycolipids. To whomever you will ask on this earth about carbohydrates, they will say the only thing which is cell recognition. Yes, because they do involve in cell to cell recognition mechanism, where these carbohydrate forms a unique cell identity tag. The best example of cell recognition are fertilization, where the sperm and egg recognize each other. First, we'll discuss what is cell recognition. So, it's the process where one cell identifies and interacts with the another cell using molecules on their surface. They are usually glycoproteins and glycolipids, as I said. Okay, let's see what their example says is. See, once the sperm and egg have specific surface protein on them that must match with each other, that ensures that the only sperm of the same species can fertilize with the egg. Suppose, that's the fact that a sperm An egg which is coated with the glycoprotein layer, which is called the zona pellucida. Don't take stress, that's in your class 12th chapter. I will explain it later. And the sperm has proteins that can especially bind to a matching protein on the egg. So only the specific sperm membrane proteins can recognize and bind with the glycoproteins on the zona pellucida. And it is important for the sperm to recognize and bind with the egg cell before the fusion can occur. And after one sperm fuses, the egg changes its shape to prevent the other sperms from binding with the egg. And don't forget, only in the same species the sperm can bind and can fertilize. And the another best example of it is blood antigens. Now see, the red blood cells have specific molecules, antigens on their surface. Okay, so these antigens act on the cell as like a ID tags, and that help the immune system to recognize a self from the non-self. You are smart enough to know what drastic can happen if someone receives the wrong blood type. Like suppose type A person gets the type B blood, their immune system recognize this type B as a foreign antigen, and they soon start triggering the immune system and they destroy the foreign RBCs. By this, the cell recognize in action and they detect the other cell based on their surface molecules or antigens. So now we are done by the fluid mosaic model by Singer and Nicholson. Now we'll look their conclusion in, we'll do it briefly in some sort of time. So the word quasi fluid, the word itself quasi says that they are partially fluid, means it refers to a semi fluid or partially fluid behavior of the phospholipid bilayer in a plasma membrane. So the membrane is not completely fluid like water or not too rigid like a solid. Other than the fluidity, the components of their membrane are not fixed at a particular place. They are always moving in some directions within the layer. Their dynamic movement gives the membrane a special kind of flexibility while maintaining its basic shape at the same time. So you can find two different types of movement in their cell membrane. One is for the protein and another is for the lipids. So the proteins show the lateral movement, which is very common to it, where the phospholipids and proteins move side to side in the layer with each other. We have this bilayer structure where the lipids are moving within the same layer. You can see in the diagram, it's clearly given there by the arrows, which shows that they are just shifting to each other's position in the same layer with the very high speed, where these lipids shows 10 million lateral movements in 1 second of time. Then we have another flip-flop movement which occurs rarely, where the lipids move from one layer to the opposite layer, where they spins around its own axis in a 360-degree angle, and their rate is low as compared to lateral movements. They are considered as rare because of their hydrophilic heads, which has to pass through the hydrophobic codes, which is energy demanding. They demand some sort of energy help. And this ability to move within the membrane is measured as its fluidity. And this fluid nature of cell membrane is also important in various functions to perform, like cell growth, formation of intercellular junctions, endocytosis, secretion, and cell division. Okay, now come and tell me, what is the first thing that comes in your mind when I say cell membrane? Is it a thick plastic-like sheet structure? No, you are wrong here. Let me tell you that they are not thick, not rigid solid membrane. They are like a thin flowing lipid bilayer structure, which is not solid like ghee but fluid like a oil. Now we will see the functions performed by the cell membrane, as I said. So first we have cell growth. See, cell growth is closely supported by the cell membrane, but it's not the primary function of cell membrane like transportation or signaling like other functions. So the cell membrane plays a supportive role in regulating growth, which is done by adding phospholipid proteins and other building block elements in their membrane through exocytosis. for their cell growth and cell division. Then we have secretion, which is a process by which the cell releases substances like hormones, enzymes, or other waste out of the cell into the extracellular space by the exocytosis. Then we have the another essential function of cell membrane, which is endocytosis, which is a process by which the cell membrane engulf materials from the outside environment and brings them into the cell by forming vesicles. Lastly, the formation of intercellular junctions is another important function of the cell membrane, especially in the multicellular organism, where these junctions are formed by the specialized membrane of proteins that help cells to stick together and communicate, coordinate, and control all the functions together. So these are all the important functions which is given in your NCERT. Other than that, let me add one or two more. So they can self-repair themselves by their own by recognizing their lipid and proteins for their minor damages. Yeah, of course. And they are responsive also. It responds to the external stimulations like hormones and temperatures. They are metabolically active, which is important for them by playing a role in process like cell signaling, endocytosis, exocytosis, and energy generation, especially in the prokaryotes. So yeah, that is it. And yeah, one more thing written in your NCERT, that they are the delimiting structure of the cell. It means they are defined in such a way that they set the boundaries of the cell to control what should go in or out of the cell. So yeah, that is it for today. Thank you everyone for being till the end of this lecture. Hope you all liked it, and soon we will come with the part 3. Till then, keep watching, keep smiling, have a good day.